am Madhurima Datta, Zoology and Biology Educational Content Creator. Now's content for B.Sc Zoology Honours, Physiology Honours, Microbiology Honours and M.Sc Zoology, Microbiology and Physiology, and also exam for CET PG exam, GATE exam, CSI NET, Life Sciences exam. Paper Evolution, Unit 1, Evidences for Evolution. Today I will discuss about the homologous organs. So, Darwin described as evolution is descent with modification. Next, concept and definition of the homologous organ. In evolutionary biology, homologous organs are structures present in different organisms that share a common ancestral origin and structural framework despite potentially serving distinct functional roles due to divergent adaptation. Next, classical definition of the homologous organ. According to Richard Owen in 1843, the same organ in different animals under every variety of form and function. Next, evolutionary definition. According to Ray Langston in 1870, structures in two or more taxa are homologous if they are derived from a common ancestral structure through the descent with modification. That is called homogeny. Next, basic concept of the homologous organ. It has the same source, that is a common origin, different appearance, perform different function, introduced by Richard Owen, similarity in basic structure pattern. Significance, common ancestry and occurrence of divergent evolution. Next, Ramen's criteria for homology. Adolf Ramen established three primary morphological criteria to identify homology across taxa. First, criterion of position, that is topological criterion. Structures occupy identical spatial relationships within the anatomical arrangement of the body plan regardless of modification in shape or size. Next, criterion of quality, that is spatial structure. Detailed microstructure, histological or developmental and subcomponents match across organism. Example, bone composition, nerve innervation and muscle attachments. Next, criterion of continuity, that is intermediate forms. The structural link between divergent organs can be traced through a continuous sequence of intermediate fossils form or ontogenic development stages. Next, classification and spectrum of homology. First, organismal and or anatomical level. Spatial homology. Homology of specific structures between two distinct species, that is human and bat wings. Next, serial homology. Iterative repetition of homologous structures along the body axis within the same organism. Example, crayfish, biramous appendages and human vertebra. Next, sexual homology. Homologous reproductive structures derived from identical embryonic primordia in different sexes, that is male penis and female clitoris derived from the genital tubercle. Next, genetic and molecular level. Similarity DNA sequence, same 64 codons specify same amino in different groups. First type is orthology. Genes in different species derived from a single ancestral gene via speciation. Example, human beta globin and chimpanzee beta globin. Next, paralogy. Genes related by duplication within a genome that subsequently diverge in function. Example, alpha globin and beta globin gene. Next, deep homology. Conservation of ancient and genetic regulatory network, that is controlling the development of morphologically non-homologous structures across disparate phyla. Example, the PAC6 or eyeless gene driving eye morphogenesis in both vertebrates and Drosophila. Next, phylogenetic homology. Similarity among plants or animals in different species. Example, man and horse. Next, molecular homology. Similarity of biomolecules such as RNA and DNA. Next, Hogg's genes or homeobox genes, a highly conserved family of transcription factor genes that regulate the anterior posterior body axis, pattern during the embryogenesis across bilateria. Example, the labial or PB or DFX genes cluster in Drosophila is homologous to the HOX-A1 and HOX-D1 cluster in vertebrate. Types of homology and their description and example. First type is phylogenetic homology. It is a structures inherited from a common ancestor in different taxa. Example, bird wing and bat wing bones. Next type is serial homology. Serial homology is repeated structures along the longitudinal axis within the same organism. Example, vertebra or legs, appendages of clusterian like crayfish. Next type is sexual homology. Structures derived from the same embryonic primordia in different sexes of the same species. Example, testes in males and ovaries in females, penis and clitoris. Next type is deep homology. Conserved genetic regulatory network driving non-homologous structures across the distant clades. Example, PAC6 genes governing eye development in both insects and mammals. Next, some short terms of homologous organ. First term is homoplasy, a character shared by a set of species but not present in their common ancestor. Includes convergence, parallelism, and reversals. It creates false signals in phylogenetic reconstruction. Next, apoplasogeny and synapomorphy. Shared derived homologous characters, that is synapomorphies, are used in cladistic to construct monophyletic taxa. Next, developmental constraint. It is highly conserved signaling pathways, example sonic hedgehog, SHH, WNT and FGF8, which maintain homologous anatomical positions despite drastically altered gene regulatory pathways in distantly related embryos. Now the topic has ended. Please subscribe my YouTube channel Biogenesis by Madhurima and follow my Facebook account Madhurima Datto for all video of graduation and masters in life sciences.